So I try to really separate this when with resveratrol, specifically talking about is it a longevity molecule versus might there be other things that resveratrol does that might be beneficial for some people. So to be very clear, in the scientific literature, the initial claims that resveratrol could increase lifespan, delay aging, have been largely disproven. It's been not reproducible, certainly in mammals, mice in the laboratory. There is no lifespan benefit from resveratrol and no evidence that resveratrol has a longevity effect in people. So it doesn't mean it couldn't have some health benefits, but essentially all of the initial work has been disproven when it comes to resveratrol as a longevity molecule. And again, people can, they're going to say, well, that's just one guy's opinion. There was a meta-analysis done, I think it was published last year, that looked at all of the published experiments on resveratrol in any model organism, and the average effect on lifespan across all of those studies was 0.
All right, Matt Kaeberlein, what are the biggest longevity scams that are out there right now? It seems like everywhere you turn, someone's trying to sell you something.
Yeah, that's a tough question. I'll say first of all that I think there's a lot of misinformation, and so I would say in the realm of supplements, that's one area where there's a lot of misinformation, some scams, some I think honest misunderstandings. I would say the direct-to-consumer biological age tests are another place where there's a lot of misinformation, some intentional. And then, I wouldn't necessarily call it a scam, but I think this sort of fixation with, very early experimental stuff like going to a secret island and getting gene therapy is certainly not a great idea and not very scientifically well thought out at this point.
So after today's video, I put a link down below for 25% off Seed's Daily Synbiotic. I'm a huge fan. It's the only probiotic that I personally use. And the interesting thing is they publish a lot of their own literature. So they publish their own clinical trials. And sometimes it doesn't work in their favor, but they publish it anyway because they are honest. And that's what I appreciate about them. But interestingly enough, it's the only probiotic that has this multi-stage delivery system where it has a capsule inside of a capsule. So when you think about probiotics, it's easy to think supplement company sham. But the reality is there is not a whole lot more in the world of supplements that is more vetted out than probiotic usage. The problem is most are destroyed in the gut. So that's what I like about Seed, is they put their money where their mouth is with their technology, with their clinical trials, and also just with overall customer service and quality products. So that link is down below for 25% off Seed's Daily Synbiotic.
Let's double click on kind of the supplements for a second. I know you and I have a little bit of a bone to pick occasionally with resveratrol.
Yeah.
Can we just kind of jump into that? What are the claims around resveratrol?
Yeah.
What's the actuality of it, and what's going on there?
Yeah. So I try to really separate this when with resveratrol, specifically talking about is it a longevity molecule versus might there be other things that resveratrol does that might be beneficial for some people. So to be very clear, in the scientific literature, the initial claims that resveratrol could increase lifespan, delay aging, have been largely disproven. It's been not reproducible, certainly in mammals, mice in the laboratory. There is no lifespan benefit from resveratrol and no evidence that resveratrol has a longevity effect in people. So it doesn't mean it couldn't have some health benefits, but essentially all of the initial work has been disproven when it comes to resveratrol as a longevity molecule. And again, people can, they're going to say, well, that's just one guy's opinion. There was a meta-analysis done, I think it was published last year, that looked at all of the published experiments on resveratrol in any model organism, and the average effect on lifespan across all of those studies was 0, meaning that for every study that was published that reported a positive effect, there was a similar study published reporting a negative effect, and this was dozens of studies. And I would say, given the bias in publication towards positive results, there are probably lots of studies that were never published that had a negative effect. So I think it's pretty clear resveratrol is not a longevity molecule. Now, I know people got very excited by the fact that it's found in red wine, and of course the idea that red wine might be good for you because of the resveratrol is very appealing. Again, no real evidence to support that in my view.
What was the mechanistic claim that resveratrol would even...
Right. And this is where I think the story gets a lot more complicated. So there's this class of proteins called sirtuins. They are named after a protein that I actually studied as a graduate student called SIR2. And it was actually my PhD thesis work that showed that if you overexpressed this protein in yeast cells, you could increase lifespan. And so David Sinclair proposed a model. They published a paper claiming that resveratrol activated this family of proteins, sirtuins, which are named after the yeast SIR2, and that's how it was affecting longevity. That turns out to also be wrong. We actually published one of the first papers back in 2004, really explaining how that initial result was a test tube artifact. So that part of the story turned out to be wrong, and then the initial reports that resveratrol could increase lifespan in mice also turned out to be not reproducible. So there are multiple places in this chain of argument that have in the long run been found to be weak links.
Are there any real connections with the SIRT pathway and longevity, or is it all fairly speculative?
So that I think still remains to be determined. I think it's clearly the case. If you think about the proteins, genes that we know affect longevity across many different animal models, there's a network there of multiple proteins talking to each other and metabolites. And sirtuins are in that network, no question about it. I sort of think of it as if there are certain nodes in that network, certain knobs that we can turn that are potent modifiers of the biology of aging. And those are the ones where in experiments we can see big effects on lifespan. It has not been the case that people have been able to robustly or consistently modify lifespan or health span by turning up or turning down sirtuins. So I think it remains to be determined how useful sirtuins are as a target for modifying the biology of aging, but they are certainly connected to that network.
I mean, and not to just take all the sizzle away from things, but I mean, the best ways to activate sirtuins, I mean, realistically, diet, exercise.
Well, that's an interesting question. I don't actually know that there's a lot of evidence to say that diet, exercise are activators of sirtuins, or that activating sirtuins in general is going to be beneficial. And again, this is where we're getting really deep in the weeds really quickly. It turns out humans have 7 sirtuins. Yeast have 5. So it's a family of proteins. We don't really know. So in yeast, we know the one that is important for longevity. The others don't really seem to be important. We don't really know in humans which of the sirtuins are important for longevity, and if any of them are potent regulators of longevity. One of the sirtuins, they all go by SIRT and then a number, SIRT1, SIRT2. SIRT6 seems like the best candidate in mammals, but it's not really clear that diet and exercise are going to activate sirtuins. Now, I think maybe where you were heading is we know for longevity, diet and exercise and sleep quality and human connection relationships are the things that have the most potent effect on health span and lifespan in people. I think still the question is, are sirtuins really important for health span and lifespan in people, or are they potent regulators of longevity?
Yeah, or is it a lever worth trying to spend money on?
I think that's the main thing.
Yeah.
And again, we haven't talked about NAD precursors yet, but again, a lot of the excitement around NAD precursors is connected to the idea that NAD is going to activate sirtuins. So as you know, NAD is sort of a cofactor for thousands probably of chemical reactions in our cells, mitochondrial function, central metabolism. It turns out that sirtuins use NAD as part of their chemical reaction to primarily take acetyl groups off of other proteins. And so if you increase NAD levels, because it's a substrate in the reaction, you'll cause the reaction to go faster. So NAD is talked about as an activator of sirtuins. There's a hypothesis that NAD levels decline with age. And so the idea is if NAD declines, sirtuin activity declines. If we could restore NAD levels with NAD precursors, that would boost sirtuins and potentially have a longevity benefit. This is, again, where the story gets very complicated. Because number one, it's not completely clear that NAD levels really decline by a lot with age or in all tissues and organs with age, although it gets talked about as if that's clear. And it's not really clear that increasing NAD levels is going to robustly activate sirtuins. It's going to do a bunch of other stuff too. And it's not really clear that activating sirtuins is going to have a positive effect on lifespan. So there's a lot of speculation, I would say, a lot of weak points in that model. Myself and many others have tried to probe the model. I mean, again, I always view our job as scientists is to try to ask, okay, where are the potential weak points in the model? And let's try to break it so that if we can break it experimentally, we can make a better model. So a lot of us have tried to probe that model and found a lot of weak points that we can break. I think I'm of the opinion I'm still waiting to. I'm reserving judgment on NAD precursors, but I don't think if boosting NAD has a positive effect on health and potentially longevity, that it's going to be primarily due to activation of sirtuins. I think it's probably other effects that NAD has on all of the other chemical reactions that it's involved in.
If we talk about longevity scams in the first place, what about NAD injections? What about, is there any evidence there? And I know we can't even say evidence for longevity.
Yeah.
But is it doing anything? I mean, some people claim that you get an energy boost from it. Quite frankly, I don't feel much when I do it.
But I haven't done it yet. I would say I'm NAD curious. Like I would give it a try. So again, I'm hesitant to call it a scam per se, because I think there are some plausible biological mechanisms by which that could be beneficial. We just kind of talked it through, right? If it is the case that NAD levels decline with age or with metabolic stress, and you could boost that NAD, that could be beneficial. And so then the question is, what's the best way to boost NAD? So I think the NAD precursors that you take orally, they will boost NAD, at least in people who have low levels of NAD. Would NAD injections actually get into cells and boost the bioavailable NAD? Yeah, it probably does. Is it a direct mechanism or indirect as the NAD gets broken down to a precursor and then resynthesized in the cells? That's all, to be determined to some extent, but it probably does boost NAD. So I think there's reason to think it could be beneficial. I don't know of any solid evidence suggesting that NAD infusions, from the perspective of randomized clinical trials, have robust positive effects. But it wouldn't surprise me if it could have beneficial effects. Could it be harmful? Yeah, it could also be harmful. Like I think we don't really know what the risk profile looks like. Probably not a high risk, but everything has risks. And if you're going to, super physiological levels with an NAD infusion, that increases the likelihood that you could cause something to go wrong simply by getting the homeostatic system out of balance by dramatically increasing this cofactor that's involved in thousands of chemical reactions. So I think we really don't know. One last thing to say, because I think a lot of people don't realize this. So there are really kind of 4 NAD precursors that people think about, orally available NAD precursors. There's nicotinamide mononucleotide, NMN, and nicotinamide riboside, NR. Those are the 2 that get the most attention sort of in the influencer world. Then there are nicotinamide and niacin. All of those, if you take them orally, essentially have the same effect on NAD. And in fact, there's some evidence that NMN and NR actually get broken down to nicotinamide and niacin in the gut, and then they get taken up that way. So the reason I'm saying this is NMN and NR cost about 100 times more than nicotinamide and niacin. So if you really, really think you want to take NAD precursors, sure, you're welcome to buy the expensive stuff that gets broken down to the cheap stuff in your gut, but there's no real good reason to do that.
If we were to look at, what do we know from the literature depletes NAD, right? So in this case, like we say, okay, we don't know if it declines with age, but are there some things that we do know that trigger a decline in it?
Yeah, I think again, this is, I'm a scientist, so I can't help thinking like a scientist, right? When you say no, I don't think there's a lot that we know with 100% certainty, and this is why I was saying it's a little bit unclear. People talk about aging as if it's a fact. Some people do, as if there's a decline in NAD levels with age. Period. Full stop. That is almost certainly not true. I think what could be the case is that in certain tissues and organs with age, we see a decline in NAD levels, at least in some people, or in some individual animals. But we don't have we don't have any great data that I know of looking this at the individual level. So it still remains to be determined if aging is causes a significant depletion of NAD. We also have to be a little bit precise in, are we talking about the NAD and NADH combined pool, or are we talking about NAD itself? So again, it's getting into the biochemistry a little bit, but NADH is the reduced form of NAD. And when we're talking about mitochondrial metabolism, for example, those convert back and forth as part of the cell's production of ATP in the mitochondria. So you can certainly have situations where the oxidative redox balance changes, so you shift so that you become depleted with NAD enriched for NADH or vice versa. So, for example, if you have mitochondrial dysfunction, what will happen is you will shift towards higher NADH, towards the reduced state, because you aren't no longer able to restore that NADH back to NAD through the electron transport chain. So that's a condition where you're functionally depleted for NAD, but you still have the same total amount of NAD plus NADH. So those kinds of situations, I would say mitochondrial dysfunction or things that lead to temporary mitochondrial dysfunction tend to perturb the NAD-NADH balance. And that's a place where NAD precursors might actually be beneficial. And there's some evidence to support the idea in animal models that have mitochondrial dysfunction. NAD precursors could have positive effects by sort of suppressing some of the consequences of that mitochondrial dysfunction that are driven by the loss of available NAD.
Yeah, or perhaps like, when you're driving ROS really high through the roof through some acute situation or something like that.
Right. So again, I think, but this also makes it where the system is much more complicated than we often give it credit for, right? And I get it. I mean, all of us, we have to kind of think in simple terms and we have to kind of think in simple models, but it's important to recognize that, NAD to NADH redox status is going to be connected to glutathione redox status, which is going to be impacted by reactive oxygen species. So all of this is kind of tied together. And so, is changing the NAD-NADH redox status going to have the biggest effect? Or maybe glutathione's better, right? Or, maybe antioxidants. So this is also why a lot of these things, when you take them as supplements, may not have certainly aren't going to have only the effect that you expect, and may have very different effects. And even if there are positive effects, it might not be for the reason that you anticipated. I think antioxidants are a good kind of example here where, people have talked about taking antioxidants as supplements for decades, right? And all the benefits of antioxidants. That's sort of a catch-all term, though, because antioxidant is really a relative term to any other molecule. Is it more reduced or less reduced, right? So these things are going to impact, many of them are going to impact the overall redox status of the cell and have unanticipated consequences, maybe on NAD biology.
What about if we pivot over to the more pharmaceutical side and we talk about the use of metformin and amplifying AMPK, kind of going that route?
Yeah,
that was a big boom there for a while, and then it kind of died away, and it seems to have these like resurgences. What's your take on that? First of all, I mean, your opinion. You don't have to give the scientific side first. You can say, what's your opinion there?
Yeah. So my personal opinion is that metformin is a really good diabetes drug. It's probably beneficial for people who have diabetes or pre-diabetes. I don't think there's much reason for non-diabetics to take metformin for putative longevity or health benefits.
Is there I've often wondered this. I mean, is there a risk of driving up AMPK and essentially suppressing mTOR in terms of muscle growth, in terms of just muscle preservation, in terms of just pro-growth signaling in the first place that might also be used for repair?
That's certainly possible. And I think with metformin, there are at least a couple of mechanisms where metformin specifically could have a negative impact on muscle growth or benefits from exercise. There have been a couple studies. Ben Miller from University of Oklahoma has done some of this work showing that metformin can have negative impacts on exercise, the benefits associated with exercise. Now, is that through AMP kinase or is it through other things that metformin does? I think is unclear. So metformin, again, we don't know exactly how it's activating AMP kinase, but I think the best evidence is that it's a mitochondrial toxin. So you're actually inhibiting mitochondrial function, and that has the effect of activating AMP kinase as a sensor of ATP and ADP, or AMP and ADP. So that's probably how metformin is activating AMP kinase, but it's a very dirty drug. It does a lot. It has a lot of targets, and so it could be other stuff that metformin is doing as well. There's also evidence. I don't know how strong it is. I haven't seen the data. I've just heard many people talk about it, that metformin reduces testosterone in men. So that could also impair muscle growth in response to resistance training. So it's a complicated molecule. I do think it is important for people to recognize that I think there's been this misperception that metformin is completely safe. It's been used in diabetics for decades. We know everything about it. There's really minimal side effects. first of all, if you're a non-diabetic using metformin, that's a different situation. The risk-reward calculation is different because you don't have diabetes. And I think we're learning that there probably are some negative effects of metformin that at least haven't been widely appreciated. I'm sure that the docs who were using metformin for diabetics knew about some of these side effects. So I don't really know the answer to your question. I'm just trying to point out that metformin has a lot of effects, and we don't really know the mechanisms, which is kind of interesting for an FDA-approved drug that's been approved for decades. We really don't know exactly how it's working. And even is all of the benefits of metformin for diabetics through AMP kinase? I don't think that's been completely determined.
Yeah. So, well, what about if we look at the opposite? We look at more the mTOR side of things. I mean, let's open up that discussion for a second, and we can kind of bring it back down to basics for a minute, because I know that people that watch this channel like the high level, but they also like the weeds. So if we go high level for a minute, I mean, how important is this balance of AMPK and mTOR, and are we overblowing it?
I don't think we're overblowing it, but I do think it's important to recognize that first of all, there's sort of natural circadian or environmental effects on mTOR and AMP kinase and the sort of metabolic network that should happen, and we need that. So it's not on off, it's not up down. It's sort of there's a cycle to these things, and there's very different tissue and organ specific ways that AMP kinase and mTOR need to be regulated. And I think oftentimes people think about Rapamycin, which is an mTOR inhibitor, as it's bad for muscle, right? Because we know you need to turn up mTOR to build muscle, right? Which is true, you do. And if you turn mTOR way down chronically, that's going to make it harder to build muscle. But Rapamycin isn't necessarily going to prevent people from building muscle because of the way it's metabolized, and it depends on the dose, and it depends on the way you take it. So, and we want mTOR activity lower in some cells and tissues. It's actually probably a good thing in the context of inflammation, for example. We know that there are many conditions, autoimmune conditions, and to some extent aging itself, where there's an increase in what we call sterile inflammation, which is your body's immune system being activated towards signals it should not be activated towards. Rapamycin is really good in that context because it turns down mTOR in the immune system and kind of blunts that chronic inflammation. If you turn it down too far in the immune system, then you become immunocompromised and become more susceptible to infection. So I just think there's a lot of nuance here, and mTOR good, mTOR bad is not going to capture that nuance, and we need to think about it a little bit more thoughtfully and critically.
Does diet influence mTOR as much as people think? So, I mean, I would say this, we talk about immune system for a moment, things like that. If someone is chronically calorie deprived and they're chronically under recovered and they're, in that state where they're basically creating a state of chronic inflammation, but they're not, the first thing that I think about is, okay, but you're not getting even carbohydrates or enough protein to stimulate mTOR. Can that influence it top down, or is it a little bit deeper than that?
So I think at the extremes, diet has a huge impact on mTOR. And again, there's not a ton of data in people, at least the kind of data that I like to see, which would be, you fast for a certain period of time and then you look at mTOR activity in different tissues and organs. For obvious reasons, we can't do biopsies on people to look at mTOR when they're fasting. But you can do that in mice. And again, it is worth saying, the effects of fasting in mice happen at a much more rapid time scale than they do in people. And it turns out if you fast a mouse for 3 days, it's going to die, right? So their metabolism is a little bit different. But I think fundamentally, at least in terms of rate, but fundamentally the biochemistry is very, very well conserved. So if you fast a mouse, even overnight, so their mTOR activity, and we can measure that biochemically, goes down dramatically just from an overnight fast compared to what it is in the fed state. So just that short-term fasting has an impact on mTOR activity, first in the liver, and then you start to see it in other tissues and organs. And if you fast them 24 hours, you basically have almost undetectable in many tissues and organs mTOR activity. So certainly diet can have a potent impact on mTOR. And I think that probably contributes to the circadian aspect of mTOR, or at least the cyclical aspect of mTOR. I don't know of a lot of good data on this, but it would not surprise me if different eating patterns had an impact on mTOR activity based on, for example, if you're doing time-restricted eating, or if you're eating all day long or eating late into night, that could have an impact on mTOR activity. Again, probably going to be in your liver first. And then I just don't think we have enough data looking at, what happens to mTOR in the muscle, what happens to mTOR in the kidney, what happens to mTOR in the brain with these sort of shorter term windows of fasting and feeding. And then there's what you eat, which obviously could also have an impact. I think initially people, the sort of dogma was mTOR is activated by branched chain amino acids, leucine in particular, more leucine circulating, higher mTOR. I think in general that's correct, but I think we've also learned that there are indirect mechanisms by which other types of caloric intake can also activate mTOR. So I think of mTOR more as a general nutrient sensor as opposed to a branched chain amino acid sensor only.
Yeah, I try to look at it like that as well, especially in the context of fasting when everything's just so sensitive to begin with. And, it's like any calories coming in, it's kind of a nutrient sense for that in that case.
Right.
I know you're a scientist and you don't want to have like a black and white response to a lot of things.
I struggle
appreciate
with that
because I'm the same way. But if you had to pick one that's more important for longevity, and I'm going to get to a point, but
Sure.
more important that people should maybe focus on. And I say focus on by, hey, like AMPK versus mTOR, right? The reason I say this is you've got people like Paul Saladino, right? He's shouting from the rooftops now that insulin is important, mTOR is important for growth, repair, for longevity, right? Then you have the other side that's, well, no, okay, you want to attenuate it. If you had to take a camp, like what side is more important?
Yeah, obviously context is dependent, but I think the simple answer is the best evidence we've got right now is that periodic inhibition of mTOR with rapamycin is the most potent way to increase lifespan and health span in animals. We don't know yet in people, but if we look in the laboratory studies, aside from caloric restriction, and we can dive into that, that's also nuanced, I don't recommend caloric restriction for people, and we could talk about why. Rapamycin is the gold standard for an intervention that slows aging, increases lifespan, improves health span in laboratory animals. So I'd say periodic inhibition of mTOR. I don't think you want to chronically inhibit mTOR all the time.
Yeah, I would agree. And I think there's a lot of people that have kind of experienced that through excessive fasting and things like that, where it's just like, okay, at a certain point, it's a serious line of diminishing return.
I do want to mention, because I do think certainly fasting will inhibit mTOR. I think of fasting and let's just say rapamycin, both interventions that are very potent, and caloric restriction and fasting we'll put in one bucket for now. Again, there's nuance there that's worth diving into, but we'll put them in one bucket for now. Both will increase lifespan in mice very potently, but they have overlapping but distinct mechanisms and biological effects. And I think of it as, rapamycin is a very clean drug biochemically. It has, as far as we know, one biochemical target and only one. Now, when you inhibit mTOR, that has lots of downstream effects. So I'm not suggesting that it doesn't do lots of things, but it's very clean. Fasting is one of the dirtiest drugs you could ever imagine. It probably changes 1 million different chemical reactions and proteins and metabolites. So fasting is going to do a lot more than rapamycin does. And I'm not saying one's better than the other. I'm just saying they're fundamentally different in terms of how they're working. And it would shock me if fasting didn't have effects through these other mechanisms that it's impacting in addition to its effects on mTOR.
Yeah, I agree. And I feel like most people look at fasting a little bit differently, right? They think fasting is like just a different form of caloric restriction, right? Like a 16-8 every day kind of thing, where they're ultimately just putting themselves in caloric restriction in the first place. Whereas, I look at fasting much more the perspective of infrequent aggressive fasts and then back to normal, right? So it's a shock to the system, big different thing. So, so if we look at, kind of how people typically would on the surface try to reduce mTOR. And I say that because people just watch YouTube and they're like, okay, cool, I'm reducing mTOR.
Yeah.
And it's an easy thing to fall into. But you mentioned caloric restriction. Why are you not a fan of caloric restriction?
So what I'm not a fan of, so first of all, let's start with the data. In mice and rats and simpler organisms, caloric restriction is the most effective in terms of magnitude of lifespan extension intervention we've got that's not genetic, not changing the genome, to increase lifespan, and it seems to improve a whole bunch of health span metrics. So there's no question that caloric restriction is a potent way to slow aging in laboratory animals. In fact, up to 60% caloric restriction gives up to a 60% increase in lifespan. So we're talking big effects. But those are laboratory animals living in a very controlled environment, and in mice and rats in particular, they're very likely to get cancer. Most people will say laboratory mice die from cancer. They certainly all die with cancer. And they aren't particularly susceptible to frailty and fractures and things like that. They don't die from those things. In people, we know that chronic caloric restriction, it's very difficult if you're restricting your calories by let's say you're starting at a 2,000 calorie diet. So you restrict by 60%, you're 800 calories a day, right? So it's very hard to do that long term. And the long term, it's very hard to get enough nutrition that you're not going to become frail, you're not going to lose bone density, you're not going to lose muscle mass. So aside from the fact that it's very difficult to do that, and I think there are some social consequences that go along with doing that and psychological consequences, I also think that because of the way that laboratory rodents age and the conditions we age them in, even if severe caloric restriction, let's say you could take care of the micronutrients and get enough protein and all of that, severe caloric restriction in people, even if it slowed biological aging, and it very well might, I think it probably does in people, there would be offsetting consequences that would cause you still to die earlier than you would if you weren't caloric restricted. That's speculation, but I think there's reason to be really careful with that kind of caloric restriction. I don't have any concerns about like short-term caloric restriction if it's part of your goal to get to a healthier weight, healthier body composition. Of course, that makes sense. But I think you want to be careful with assuming that just because it makes a mouse live 60% longer, I should do this.
No, I'm 100% with you. And I feel like, there's a fine line between, okay, yes, if you're reducing your fat mass by caloric restriction, then sure, like that makes sense. But This is a means to an end. That's, I don't know.
And you see this a lot in the longevity field, right? People just sort of throw this tag longevity on there. And again, I get that people want the simple answer, but there's a lot of important nuance that needs to be talked about. Otherwise, people are misled and they think, oh, I should just do this because it's a longevity protocol, right?
Is there any merit to so I'm a big fan of eating more and moving more, right? That's like how, if I'm going to induce a deficit through my daily life, I would try to have it come from, okay, I'm consuming a fair bit of food, but I'm also just moving a lot, right? I'm gonna move at a greater flux, so to speak. But, I come up against resistance sometimes on that, saying like, okay, well, you're creating a lot of exhaust in your body. That's just driving up ROS so high by moving more. Like, right. I mean, but what's the alternative? Like, just like starving myself and not moving very much? I mean, what's your take on that? Like, does excess activity, I mean, not even excess, where do we draw the line, right? Like, is someone that's eating more and moving more, are they taking years off their life by doing that?
Yeah, I mean, again, the honest answer is we really don't know for sure. I think the sort of practical answer is that, first principles, look at body composition and body weight. That's probably going to be more important than how you get there. Mm-hmm. That makes sense. So what I can say from the laboratory studies of the biology of aging is there is a theory, this kind of rate of living theory, that metabolic rate is a predictor of longevity, and this idea of producing more ROS, make you age more rapidly. That theory, I think there are correlations when you look across the animal kingdom, but when you start looking at individuals within a species, I don't think there's much evidence to support that idea that a higher metabolic rate is going to lead to faster aging at the individual level. So I don't think there's any great evidence to suggest one way or the other. I would say from a pragmatic perspective, for individuals, if that strategy works for you to maintain a healthy body composition and body weight, great. That's better than, the alternative, which is not maintaining a healthy body composition and body weight. And is it better to get there by restricting calories or by exercising more? Probably some of both. And I would also say, I think what you eat matters. So, if you are at a healthy body composition and body weight for you and you're eating a relatively clean diet, you're in a pretty good place. Like, I don't know that I would worry so much about exactly how you're getting there.
Yeah, no, it's a great segue into biological age as well, because you mentioned that. You mentioned, okay, like, hey, you might be able to move the needle a little bit on this biological age, right? But then you're talking about like real life, like frailty, you're talking about muscle mass. So like, it's a great segue to talk about that.
Sure.
what is really the problem with biological age? I mean, I could think of a few, but I'll ask you.
Well, I mean, I think one of the problems is there's not consensus on how we define biological age fundamentally, even among the scientific community. So that leads to a lot of confusion, right? So I think conceptually, what we mean by biological age, and I think everybody agrees conceptually about what we mean by biological age. Different individuals age biologically at different rates, and different animals age biologically at different rates. And to give an example, I think everybody can understand, you can think about dogs, right? We've all heard this idea that 1 human year is about 7 dog years. Well, that's not true for chronological time, right? Your poodle is 1 year old after 1 year. But what we really mean is that biologically, dogs are aging about 7 times faster than people are. They are getting the functional declines, the diseases of old age, about 7 times faster than people are. And again, that's not perfect. It's not linear. It matters on the size of the dog. But conceptually, I think we all get the idea that dogs age faster biologically than people do. But chronological time doesn't change. So that's the difference between chronological age, number of candles on your birthday cake, versus biological age, how old your cells, your tissues, your organs are. And how do we determine how old they are biologically? It's based on function. And this is where it starts to get into a little bit of a circular argument. So then when we look at function, that really is telling us about health. So is biological age different from health? Are they 100% the same? They're not identical. We can all think of ways that we could trash our health without necessarily improving or decreasing our biological age. So again, the concepts are overlapping, but they're not identical. And there's a lot of confusion. And then there are lots of things that change with age, right? With biological age. So hair color in many people, hair loss, wrinkles on the skin. So these external cosmetic things we all see. There are things inside our body that are changing as well. And we can measure some of those, and we can look at them functionally, like what's your VO2 max? That on average in most people, all people actually, is going to go down with age. We can measure epigenetic changes. So methylation is what people are usually using, methylation of the genome. That changes in a predictable way with age. But there are tens of thousands of things we can measure that we can then correlate with chronological age, number of candles on your birthday cake, or with health, or with mortality risk. And so what people have done is they picked subsets of features. So let's just talk about the epigenetic, the methylation test, because that's what most people are going to be seeing if they see a biological age test, right? What this does, there are hundreds of thousands of different places in our genomes that can have a chemical modification called methylation. And that can be sort of an on-off, so it's binary. It's either methylated or it's not at that position. And what you can do is you can find subsets of these marks, let's say 100 of them, that correlate really well with chronological age, with how many birthdays you've had. And so those are chronological age clocks, and we can measure that, and we can do that really well. Like, it's very precise. Those are the first generation, what people called aging clocks. And then what people did was to say, if we look at that data, most of the people are right along the line where their methylation chronological age is matched with their real chronological age, because we know how long they've been alive. But some people are above the line and some people are below the line. And those people might be biologically aging at a different rate. And so then people found different signatures within the methylation pattern that are associated with your risk of dying in 3 years or 5 years or 10 years. And the way this is done is with what are called longitudinal sampling. So there are these longitudinal studies of aging where people collected blood 20 years ago, 10 years ago, 5 years ago, and they're in a biobank. And many of those people have died. So we now can go back to those samples, measure their methylation pattern, and identify new patterns that are highly correlated with the people who died in 3 years or 5 years or 10 years and the people who didn't. Those now are thought to be, or at least talked about as, biological aging clocks, because mortality risk, your risk of dying, is highly correlated with your biological age. And it's also correlated with your chronological age. So I know this is complicated, but it's good for people, I think, to really understand what these tests are measuring and recognize it's a correlation of a correlation of a correlation. It doesn't mean there's no value there, but it's not completely clear that this is really measuring biological age. And so you can do this for all sorts of different things that might be interesting to look at, like what's your risk of getting cancer? What's your risk of getting heart disease? What's your risk of getting kidney failure? And so people have generated even newer clocks that are correlated with your risk of individual diseases or all diseases altogether or other functional declines. And those are like the third generation biological age clocks. And I'll just finish up. I think what's really interesting is you can also find correlations that are associated with the rate of change. Now, again, going back to these longitudinal samples where they've been collected over 30 years, you can see which people had declines in health rapidly and which people sort of stayed really healthy over that whole time. And you can find different patterns that are correlated with that rate of change. Those are these aging rate clocks. So Dunedin pace is the one that, again, if people have heard of these, that's the one they might have heard of. Which are maybe predictors of your rate of aging, but we don't really know. So that's kind of the nature of these biological age tests. It's also worth saying there's nothing magic about the epigenome. So epigenetic changes are one of what we call the 12 hallmarks of aging. So it's part of biological aging, but it's not the whole picture. And you can create similar clocks with all sorts of other types of data. In fact, you can create clocks that are almost as good from facial imaging. So you can take pictures of people of different ages, and AI can identify features of those pictures that are strongly correlated with chronological age. If you know about those people's health status, AI can find features in the facial images that are predictive of health status. It's the same idea. That's exactly what we're doing with the methylation. It's just a different type of data. You're finding a subset of that data that's highly correlated with your feature of interest. You can do it with blood work as well. So there are blood-based, like clinical chemistry, biological age predictors. So I think the take-home message is all of these things are super useful from a research perspective, and they're getting better. And they're going to actually, I think, in the next few years, be very powerful for helping us not only understand the biology of aging better, but maybe predict which drugs could have a positive effect on aging in people. And if they work really well, do it in an individual way where maybe rapamycin is great for you, but it's not great for me. And you could have a test that would tell you with these biological age predictors whether it's moving the needle in the right direction. But we're not there yet. And I think where I have real concerns is with the direct-to-consumer biological age tests. I'll just say, I did a self-experiment. I tested 4 different companies. I got 2 tests from each company on the same day from the same biological sample. I sent those tests back in. The numbers were all over the place. So I was 53 at the time I did it. Some of them came back at 42. Some of them came back at 63, converged on my chronological age. But as a scientist that knows something about statistics, I look at that, I'm like, this is garbage. This is all noise. There is no utility here at all. And so I have real concerns that there's really no quality control. And it's not the methodology. As I said, these are useful research tools. I think it's the way it's being applied in the direct-to-consumer sphere. And unfortunately, a lot of people are getting misled into thinking that this is telling them something important about their health. So I have real concerns about that aspect of the field right now. And again, it's worth just saying, however you feel about the FDA, none of these tests have been evaluated or approved by the FDA. So it is the case that you and I could decide right now, we're going to make up a test for whatever we want to make it up for. We can put it in a kit, make it look really nice, and sell it. Yep. So it's really unfortunate, I think, that there isn't a higher standard, and I would argue a little bit more regulation so that at least people aren't being deceived by the way these things are being marketed.
Yeah, it's very sloppy. It's really sloppy. And it was a rush to, we saw like a big, 4 or 5 years ago, kind of a big increase in awareness of it. And since then, it's like they're just popping up everywhere.
Yeah. And I think, there are some parallels. I think this is a different discussion, but I think you could make some of the same arguments about the microbiome tests, right? No matter how you feel about the microbiome, again, microbiome, super important biology. As we learn more about it, it's going to turn out to be really, really, really valuable. But many of these direct-to-consumer microbiome tests I would put in that same sort of category. There's not quality control. Even if the data itself is informative, we don't have any information on the precision or accuracy of the tests as they're being manufactured and sold to consumers. So it's a challenging time, and I think people sort of assume that if a company is out there and they say this is a diagnostic test for X, right, biological age, that it must be, and there's no quality control.
Yeah. No, there's no line at all. What, if it even matters at all, what can people do? What should they look at? What should they test?
Yeah.
What do they pay attention to?
I mean, I'm a pretty pragmatic guy, believe it or not, and I'm a big believer in function, right? I think looking at function and some biomarker assessments, and we can talk about the specific biomarkers that I think about. But those are the things that I would really prioritize. So by function, I mean things like VO2 max, or how much weight can you lift or move, or can you do depending on where you're at, can you do the daily activities that you want to do, right? We can look at there are more sophisticated tests to look at cardiac function, kidney function, liver function. I think if your organs and tissues and your body is functioning well, you're probably in pretty good health. Is it perfect? No, it's not perfect, but it's probably about the best we can do. And for quality of life, again, people sometimes have this question about, longevity versus health span, and we don't want to increase longevity without increasing health span. First of all, I think that's sort of a non-argument, and we could talk more about that if you want to. But really, for quality of life, for health span, it's function and being able to do what you want to do when you want to do it that's most important. So those are the kinds of things I think about. I would also suggest, though, that taking a proactive approach is really important, right? So our current healthcare system is very reactive. If you're going to a typical primary care doctor, you're probably getting very basic blood work, and typically they're not going to do anything until you're sick. I think, globally we need to change the system, but locally at the individual level, there are a lot of things that people can do as individuals to be more proactive about their health. And so, these are the things where I would think about some of the advanced blood work that's available, imaging, point of care ultrasound is an example, knowing your plaque burden. There are tests that can do that. And even, again, I'm not trying to sell whole body MRI. That's kind of the high end of the spectrum, but that's an example of a test where, depending on the person and your own willingness to, deal with information, right? Like you might find something, and I know that creates anxiety for some people, but if you're one of those people like me, where I want the data and then I can act on it, I want to know. So whole body MRI is an example where you can find things early and you can take action. But even things simple like wearing a continuous glucose monitor, I think that needs to be done in a way where people are getting good education about what they're actually seeing. But if that's done under the supervision of a good doc, that's really powerful. You can learn about how your body responds to the food you're eating, and you can't really unlearn that once you've seen it. And I think those kinds of things can be quite powerful. Many people will find, maybe not the people watching your podcast, but if we just take a cross-section of America, a lot of people, if they put on a CGM, they're going to learn real fast they're pre-diabetic or diabetic, and they didn't know it, right? And then they can do something about it. So those kinds of things I think are really important and powerful ways for people to actually take proactive steps for their health.
Yeah, I totally agree. It frustrates me a little bit that so many people bag on CGMs for non-diabetics. When in reality, that's what I've always said. Is they only need to wear it for a couple of months to really have a lot of answers.
It's a learning tool, yeah.
No one's suggesting you're wearing this thing forever.
Right.
But I mean, if you have an opportunity over the counter to get a CGM and learn for yourself so that you can take that to your PCP and say, hey.
Although I will say, there's a couple things I would say on that. One is, I think it depends on the person, right? There are probably, again, with your audience, there are probably a lot of people who can do it themselves and they'll understand what they're seeing. I do know many people who have tried CGM and have not gotten guidance on what it means. And I think it can derail people sometimes if they aren't given the appropriate guidance.
For sure.
And then the other thing I would say is not everybody has a PCP where if you come to them with your CGM profile, they're gonna even want to see it, right? Yeah. So this is where having a doc who you trust and you can work with and who will actually help you is really important.
Totally. Well, Matt, can you tell us a little bit about OptiSpan and where everyone can find you as well?
Sure. Yeah. So OptiSpan, I would characterize us as primarily a healthcare technology company. Our sort of overarching mission is to enable health optimization for as many people as possible. And I get it. That's a big sort of statement. But what I mean by that is, again, we were just talking about some of these proactive, preventative steps, this shift from, as Peter Attia would say, medicine 2.0 to medicine 3.0. What can we do in that space to have an impact and help? And so we're really trying to develop a technology platform that is scalable. But in order to do that, we have a couple of clinical programs. So we have a high-end concierge clinic that is doing things like whole body MRI, comprehensive blood work, point of care ultrasound, VO2 max testing, things like that to really take a personalized, proactive approach to health optimization or getting people on a better health span trajectory is maybe a better way to frame it. And then we also have a corporate program. And our concept there is we think there's a lot we can do. Like, what's the 80-20? If you could do everything, what's the 80-20, right? Where you can get 80% of the benefit for 20% of the cost in this case. So we think there's a lot we can do at a price point that companies would be willing to invest in their employees. And so we have been working with a company in the Seattle area running a sort of corporate health span program. And it's a truly medical program. So it's not a typical corporate wellness program. And we've learned a lot, and I think we've made a lot of progress. And our goal is to get to the point where we can really scale this to a much larger population of people. So it's a learning process. We're not there yet, but I feel really excited about the direction that we're going.
Totally, man. And where can everyone find you?
Yeah, so our website is optispan.life. So definitely check that out. And we have our own podcast called the Optispan Podcast, which is a lot of fun. And we have a lot of diverse guests on there. We do some deep dives into the science of longevity and then also cover a lot that is hopefully more pragmatic and practical approaches that people can take.
Right on, my man. Thanks, brother.
Thank you.